Hi friends, welcome back to my channel Only Bio, presenting up the important points from the chapter Genes. The terms or the points discussed here are derived from MCQs that have appeared in the various national or the state level competitive exams. Questions are formed from these terms or points either in similar or modified forms. These are beneficial as they will help you to master the important topics in limited span of time, frame expected questions, do effective revision, and make your own synopsis. The more MCQs you solve, the better you understand and get confident. So a daily practice of few minutes with me on my channel can certainly help you to master the topic. So stay tuned. So presenting up the important points from the chapter Genes from 21 to 40. Friends, I hope you're following the series. There are many more series which have been already uploaded. So it's my request to you all to go through the playlist and get ready. Keep studying for NEET, KVPY, Olympiad, your board exams. Point number 21. Each nucleosome has a diameter of about 11 nanometers and a height of about 5 to 7 nanometers. Next, a nucleosome consists of a histone octamer formed by two each of H2A, H2B, H3 and H4 histones. Next, the octamer is a disc-like core particle with a diameter of 10 nanometers and a height of 5.5 nanometers. Next, wound around this octamer core are 1 3/4 rounds of a DNA strand. The wound portion of DNA has about 200 base pairs. There is a common point from where the DNA strand enters and moves out after coiling, and this point is sealed by a single molecule of H1 histone. On the screen, you can see how exactly the nucleosome looks like. So it has a basic octamer core, that is a histone octamer core, around which the DNA takes 1 3/4 turn, and the remaining 1/4 part is nothing but the linker DNA which is connecting the two adjacent nucleosomes with each other. Please zoom in and look onto the pic. Next, the structure of nucleosome is therefore comparable to a solenoid. Next, the DNA strand after emerging from one nucleosome will move towards the next nearby. This unwound strand of DNA between the two successive nucleosomes is termed as the linker DNA. Next, in the living condition, the chains of the nucleosome are further folded to form solenoid superstructures with a diameter of 30 nanometers. Such a superstructure is considered to have 6 solenoids per turn. This kind of arrangement makes it possible to accommodate a very long chain of DNA within a limited space. On the right hand side, you can see a picture of a nucleosome individually and how they get packaged together to form a solenoid. Point number 29. The long thread-like DNA molecule consists of two strands that are joined to one another all along their length. Each strand is a polymer made up of repeated subunits called as nucleotides. Hence, each strand is called as a polynucleotide. A nucleotide is a complex of three molecules that are linked together by dehydration synthesis, that is, a water molecule is removed from each join to achieve the linkage. The three molecules joining to form a nucleotide are first a deoxyribose sugar in case of course, deoxyribonucleotide. If in case of ribonucleotide, that is in case of RNA, we can call it as a ribose sugar. Second, a phosphoric acid, and third, a nitrogen containing basic substance called as the nitrogenous base. Point number 32. A nucleotide with the deoxyribose sugar is called as deoxyribose nucleotide. Next, the names of the two nucleic acids themselves are derived from the type of sugar that they contain. While the sugar is deoxyribose in DNA, it is ribose in RNA. Next, the deoxyribose is a pentose sugar having 5 carbon atoms. Hence, we call it as a pentose sugar. Its empirical formula is C5H10O4. It has got a 5-member ring formed by 1 oxygen atom and 4 carbon atoms. You can look on the screen to see how exactly the deoxyribose molecule is assembled. There are side branches from all carbon atoms, some of them terminating in hydrogen atoms, others in hydroxyl group, and one in a CH2OH group. You can see it on the screen very clearly. Next, each group occupies a particular position. To indicate these positions, the carbon atoms are named 1 to 5 in clockwise direction, starting with the one at the side of the oxygen atom at the extreme right of the ring. Point number 37. The pentose sugar in DNA is termed as deoxyribose because it has 1 oxygen atom less when compared to the ribose sugar of RNA. In ribose sugar, the side chain of 2 carbon is OH instead of H. Look on the screen, you can see deoxyribose and ribose being drawn simultaneously for you to understand the difference. Point number 38. The sugar molecule occupies the central position in the nucleotide. To its one side at 1 carbon position instead of OH is attached the nitrogenous base, while on its other side at 5 carbon instead of OH is attached the phosphate group. Point number 39. The linkage of 2 nucleotides is necessary for the formation of polynucleotide, and it is also achieved by the sugar molecule. The phosphate of the adjacent nucleotide gets attached at the 3rd carbon position, also at the expense of the OH group. So on the right hand side of the screen, you can easily see one of the strands being drawn there. Point number 40. Within the nucleotide, the phosphoric acid is joined with the sugar at the 5 carbon position, while the phosphoric acid of one nucleotide is joined with the next nucleotide at the 3 carbon position of the latter's sugar. The phosphoric acid gives up a hydrogen atom at both these linkages during the dehydration synthesis and becomes a phosphate group, that is PO4. Friends, I'm sure this series will help you, so please stay tuned and please subscribe if you're new to the channel. Thank you.